Imagine, for a moment, the immense relief after a long battle with cancer. The surgery is declared a success, the scans come back clear, the doctors say they got it all. The war, it seems, is finally over. Life slowly inches back towards normal. But then, months or sometimes years later, a shadow returns. The cancer is back, often more aggressive, more resistant, and sometimes in a completely new part of the body. It's a devastating turn that leaves everyone asking the same crushing question: how? We thought the enemy was defeated. The answer lies with the villain that conventional treatments often miss. They're not the rank-and-file soldiers of the tumor. They are its special forces. A tiny, elite population of cells that can survive the onslaught of chemo and radiation by going dormant, like sleeper agents. These are the cancer stem cells, the hidden seeds of recurrence, waiting patiently to reawaken and rebuild their entire deadly empire. For decades, medicine has struggled to fight an enemy it couldn't fully see. But what if we could finally unmask these hidden commanders and teach our own bodies to hunt them down for good?
It's a truly compelling question. And what you've described, it really touches upon a profound paradigm shift happening in oncology right now. For a long time, our strategy is primarily focused on, broadly attacking the visible cancer cells, almost like trying to clear a forest just by cutting down all the trees you can see. But these sleeper agents, the cancer stem cells you mentioned, well, they've proven remarkably adept at surviving those conventional assaults. The truly exciting frontier now is the emergence of advanced nanovaccines. And these aren't just incremental improvements, they represent a highly promising targeted strategy to specifically identify and eliminate these elusive cells. The aim is really preventing that devastating post-surgical recurrence and the spread of cancer, metastasis. It's about not just felling the trees, but really eradicating the seeds.
And that's precisely what we're going to dive into today. How the cutting-edge worlds of nanotechnology and immunology are colliding, really, in a revolutionary way to create these next-generation nanovaccines. We'll explore the intricate bioengineering behind them, from leveraging tiny RNA molecules that act almost like genetic blueprints to precisely presenting the cancer's unique face to our immune system. You'll discover how these miniature marvels are designed, how they equip our body's own defense system, and how groundbreaking research, like the innovative work emerging from institutions such as Zhejiang University in China, is accelerating these breakthroughs, bringing us closer to a future where we can truly teach our own bodies to hunt down cancer for good, permanently disarming those hidden seeds of recurrence. Okay, let's really unpack this concept of cancer stem cells, because it's absolutely central to this new approach to cancer treatment. For so long, our focus in cancer therapy was much like a conventional war. We aimed to shrink the main tumor, reduce its size, eliminate as many cancer cells as possible. But it turns out just shrinking the tumor isn't enough, is it?
You've hit on the core problem there. Exactly. What we've come to understand is that shrinking the main tumor, while yes, it's important, it doesn't always guarantee a lasting victory. Cancer stem cells, or CSCs, are a very small, often minuscule subpopulation within a tumor. But don't let their size fool you. They possess these unique, formidable stem-like properties. Imagine them as the true architects, maybe the master strategists, or even the queen bee of the colony. They have this incredible ability to self-renew indefinitely, meaning they can make endless copies of themselves and also differentiate or sort of morph into all the various specialized cancer cell types that actually make up the bulk of the visible tumor. They're the ones who can essentially rebuild the entire destructive structure from scratch, even if, say, 99% of the regular cancer cells are wiped out.
So they're like the elite commando unit that survives the initial bombing, ready to regroup. That's quite a scary thought.
Precisely. And that's why they're so problematic. These cancer stem cells are inherently resistant to many conventional therapies we've relied on, like chemotherapy and radiation. This isn't accidental, it's a built-in survival mechanism. They can enter a dormant state, almost a kind of suspended animation, much like those sleeper agents we talked about. This allows them to effectively weather the storm of aggressive treatments. Once the drug is out of their system or the radiation effects fade, they reawaken. And when they do, they become the driving force behind tumor recurrence, the cancer coming back, and metastasis, which is the cancer spreading to new parts of the body. They are the core reason why many patients experience a devastating relapse.
So we've learned this the hard way in the clinic. Do we have tangible evidence that these specific cells are the culprits behind recurrence?
Absolutely. We've seen this devastating pattern play out repeatedly in clinical settings, providing, undeniable evidence of the CSCs' role. For instance, in bladder cancer, numerous studies have shown a clear and significant correlation between the presence and expression of certain CSC markers. These are specific proteins or genetic signals on these cells, like SOX2 and ALDH1, and a much poorer prognosis for patients. And this isn't just a slight increase in risk. It's specifically tied to how long patients remain recurrence-free after treatment and whether they develop distant metastases. It's a direct indicator of aggressive disease.
Wow.
Yeah. And other markers such as crypto-1 and DCLK1 also show strong, undeniable associations with increased recurrence and the spread of cancer. So it's abundantly clear. These cancer stem cells are not mere bystanders in the disease process. They are central to the cancer's most aggressive and dangerous behaviors. They are, quite literally, the reason the enemy often returns with a vengeance.
So this new understanding fundamentally changes how we think about fighting cancer then. If these cells are truly the hidden source of recurrence, the strategic command center, then just broadly hitting the main tumor with conventional treatments, well, it isn't enough to secure a lasting victory. It's like engaging the main army, but leaving the special forces untouched, allowing them to rebuild. We need a way to specifically identify and take out these commanders.
Exactly. This understanding has quite literally led to a complete paradigm shift in how we approach cancer therapy, moving beyond just visible tumor shrinkage. The new mission, the overarching goal, is to specifically identify, target, and eliminate these cancer stem cells. The hope is that by doing so, we can achieve truly durable, long-term responses for patients and prevent the cancer from ever coming back. And this is precisely where the remarkable capabilities of our own immune system, when given some very high-tech assistance, step in. It's about teaching our body to become a precision hunter rather than just a blunt instrument.
Okay, let's talk about our body's incredible built-in defense system. Before we even get to nanovaccines and how they give our immune system this high-tech upgrade, how does our immune system normally recognize and respond to a threat, like a rogue cell, say a cancer cell? It's not like these cells wave a tiny red flag saying, attack me.
Yeah.
Right. How does our body differentiate a normal cell from a dangerous one?
That's a fantastic question because it's an incredibly intricate, elegant process, often referred to as antigen processing and presentation, or APP. It's absolutely critical for effective anti-tumor immunity, but it is complex. Think of it like this. Your body's immune cells, especially these highly specialized ones called cytotoxic T cells, are like highly trained snipers, incredibly precise in what they target. But as you said, they can't shoot what they can't see, and they can't see the whole cell. They need a very specific signal. The key to them seeing is through MHC Class I molecules. Imagine these as little display cases or maybe billboards found on almost every single cell in your body. Their job is to constantly present tiny pieces of proteins called peptides from inside the cell to passing T cells. It's their way of showing what's going on internally.
Right, like a status update.
Exactly.
Yeah.
Specifically, these MHC Class I molecules, which in humans are known as human leukocyte antigen or HLA, like HLA-A, HLA-B, and HLA-C, these are the billboards that CD8 cytotoxic T cells read. These CD8 T cells are your body's direct killers. Their job is to eliminate infected or cancerous cells by recognizing these specific peptide fragments. MHCI molecules are quite particular. They prefer to bind short peptides, usually between 8 to 11 amino acids long, like a very specific key fitting into a very specific lock. Now, what makes vaccine design tricky here is that these HLA genes are incredibly polymorphic.
Polymorphic.
Yeah, it means everyone has slightly different versions of them, maybe millions of different variations across the human population. And that impacts exactly what peptides they can bind and display effectively. It's why a vaccine that works for one person might need tweaking for another. Personalization becomes key.
Got it. So every cell is constantly showing off bits of what's inside it to these T cells. But how do those tiny bits get selected and prepared for display? It sounds like there's a complex cellular kitchen involved.
There absolutely is a complex multi-step assembly line within the cell that prepares these messages. We're talking about the processing of endogenous proteins, those proteins made right there within the cell itself. It all starts with proteosomes. These are large, barrel-shaped enzymes. Think of them as the cell's internal shredders or maybe recycling centers. Their main job is to break down misfolded, damaged, or otherwise aberrant proteins into smaller peptides. It's a vital quality control step. If a protein isn't right, it gets chopped up.
Makes sense.
What's particularly clever is that there are different types of proteosomes, including a special immunoproteasome that can be activated by signals like interferon gamma, often produced during an immune response. When this immunoproteasome is active, it leads to different cleavage specificities, meaning it cuts proteins in slightly different places, which ultimately shapes which peptides are produced and thus what messages are displayed.
Okay, so shredding happens, then what?
Once these tiny peptides are generated in the cell's cytoplasm, they need to get into the endoplasmic reticulum, or ER. That's like the cell's internal transport and protein folding network. This crucial job falls to the transporter associated with antigen processing, or TAP. TAP acts like a specialized pump, specifically pulling peptides from the cytoplasm into the ER. It has preferences, generally moving peptides that are 9 to 16 residues long, though it can move longer ones, just maybe with lower efficiency. It's also quite selective about the amino acids at both ends of the peptide. For instance, on the C-terminal end, it tends to favor aromatic, hydrophobic, or positively charged amino acids like phenylalanine, tyrosine, arginine, or leucine. Ensures only certain peptides get through.
So it's a very selective gatekeeper.
Very selective. And inside the ER, even more precision trimming occurs. Other enzymes, specifically ER aminopeptidases, or ERP1 and ERP2, act as molecular barbers. Their job is to trim the N-terminal ends of these peptides down to the perfect 8 to 11 mer length, ensuring they fit perfectly into the MHC groove like a custom-tailored suit. Once they reach this optimal size, they undergo structural changes that prevent ERAP1 from trimming them any further. It's very neat.
Wow.
And the entire process is incredibly well coordinated by a multi-subunit complex called the peptide loading complex, PLC. This complex includes vital proteins like TAPACin and TAPBPR, which work together as a quality control team. They ensure that the MHC molecule is properly folded and stably loaded with a high-affinity peptide before it leaves the ER. It's a rigorous assembly and quality check. Only after this entire sequence, from proteasomal breakdown through TAP transport, ERA priming, and PLC coordination, is the perfectly loaded MHC peptide complex transported through the Golgi apparatus and then to the cell surface. Only then is it ready for scrutiny by T cells. This defines what messages are naturally presented to the immune system.
Wow, that is an incredibly sophisticated process. So it's like a highly choreographed microscopic dance to get just the right message onto the cell surface for inspection. But here's the puzzle. If cancer cells are fundamentally part of our own body, how do T cells, which are designed to distinguish between self and non-self, learn to recognize them as a threat? It sounds like they would just ignore them since they originated from our own cells.
That's where Krauss presentation comes in, and this is where the story gets really interesting and truly vital for cancer immunotherapy. You see, normally MHC Class I presents endogenous proteins.
Proteins made inside the cell.
Right, proteins made inside the cell to CD8 T cells, leading to the destruction of infected cells. On the other hand, MHC Class II usually presents exogenous proteins.
Taken from outside.
Exactly, proteins taken from outside the cell to CD4 T cells, which are more involved in coordinating the broader immune response. The problem is that tumor cells themselves are often quite poor at directly priming an effective CD8 T cell response. They're often invisible or actively suppressive to the T cells that could kill them, making them very difficult targets.
So they hide.
In effect, yes. This is where specialized antigen-presenting cells, particularly dendritic cells, DCs, become the unsung heroes of the story. Dendritic cells have a unique and fascinating ability. They can take up exogenous proteins, for instance, antigens released from dying tumor cells or even components of live tumor cells, and contrary to the usual rule, process them and present them on MHC Class I molecules. This is what we call cross-presentation. It's like they act as the immune system's intelligence gatherers, picking up fragments of the enemy and showing them to the snipers.
Ah, okay, so they bridge that gap. How did they get these antigens?
Dendritic cells are truly voracious. They can ingest antigens from various sources, including dying cells, through processes like macropinocytosis, which is like gulping fluid, endocytosis, taking in particles, or phagocytosis, engulfing larger bits. They can even do something called trochocytosis, where they literally nibble bits off live cells.
Nibble? Really?
Really?
Yeah.
Once a DC has acquired these tumor antigens, they process them. Now, while some pathways exist that bypass the proteasome, the ideal cross-presentation pathway for cancer mimics the natural processing machinery of a tumor cell. This ensures that the T cells primed by the DCs can effectively recognize and attack actual tumor cells once they encounter them in the body. Once DCs acquire and process these tumor antigens, they then travel to lymphoid tissues, places like lymph nodes, our body's training grounds for immune cells, for prime-naive T cells. This leads to clonal expansion and the development of a huge army of cytotoxic T cells, now specifically armed and ready to fight that cancer. It's a crucial step in turning a blind spot into a precision strike.
So the ultimate goal here is to get our immune system to truly see and powerfully attack these cancer cells, especially those incredibly tricky stem cells that cause so much recurrence. This sounds like a job for a vaccine, but for cancer, it's a bit different than, say, a flu shot, right? We're not preventing an infection. We're trying to cure an existing disease or prevent its return.
Indeed, it is fundamentally different. Traditionally, cancer therapy has heavily relied on surgical removal of tumors, followed by systemic treatments like chemotherapy and radiotherapy. While these methods have certainly made progress and saved countless lives, they often have significant limitations in terms of specificity and efficacy. They can lead to debilitating side effects because they don't differentiate well enough between rapidly dividing healthy cells and cancerous cells. And, as we discussed, they frequently fail to address the root cause of recurrence, those elusive cancer stem cells. This is precisely why immunotherapies, and cancer vaccines in particular, represent a completely new and exciting paradigm. They work by harnessing the immense inherent power of our own immune system to precisely target and eliminate cancer cells, often with far fewer systemic side effects. It's like turning our body's own defense system into a highly intelligent, self-sustaining army.
So this is where nanotechnology steps in. How does something so tiny make such a huge difference?
This is truly where nanovaccines shine. They are an emerging cutting-edge strategy that employs nanotechnology engineering at the scale of atoms and molecules to dramatically enhance the immune system's ability to respond to tumor antigens. Nanotechnology offers an incredibly versatile suite of tools designed to overcome the traditional drawbacks that have plagued cancer vaccines. Historically, these challenges have included difficulties in optimally delivering antigens to the right place in the body, the formidable hurdle of overcoming the immune suppression that often exists within the tumor microenvironment itself.
That defensive shield you mentioned.
Exactly. And the fundamental need to elicit a robust, sustained, and highly specific T-cell immune response. Furthermore, nanotechnology can help address the variable nature of individual patient immune responses and the sheer complexity involved in creating personalized treatments.
You mentioned nanocarriers. Are these like miniature armored vehicles for the vaccine components? What advantages do they offer over just injecting the antigens directly?
That's a perfect analogy. Nanocarriers, or NCs, are incredibly sophisticated engineered vehicles typically composed of materials like lipids, think tiny fat bubbles, polymers like complex plastics, or various inorganic compounds. They don't just carry cargo, they act as highly intelligent delivery systems for the antigens, the cancer's fingerprints, and for immune-boosting agents called adjuvants.
Adjuvants.
Yeah, adjuvants are like the immune system's alarm bells. They directly stimulate a robust immune response and help overcome the tumor's cunning immunosuppressive tactics. So the advantages of these nanocarriers are profound. First, they can protect antigens from degradation, ensuring these delicate biological messages remain intact until they reach their target, rather than being broken down prematurely in the bloodstream.
Okay, protection. What else?
Second, they facilitate the controlled release of their cargo. This is vital for sustained immune stimulation. Instead of a quick burst, they can release antigens over time, continuously training the immune system. Crucially, they promote targeted delivery to specific cells or tissues, which is especially important for reaching those critical immune cells, like dendritic cells that we talked about. This minimizes off-target effects and maximizes efficiency.
Makes sense. Targeted delivery.
And finally, their surfaces can be modified in clever ways, allowing for the co-delivery of adjuvant molecules directly to these immune cells. It's like sending in a specialized, highly protected rapid response unit right to where it's needed most, with a built-in megaphone to alert the troops. Without these nanocarriers, antigens might just float aimlessly, get destroyed, or fail to properly activate the immune system.
This sounds incredibly precise, like we're talking about molecular-level engineering on a grand scale.
Yeah.
How exactly do scientists engineer these tiny nanovaccines to do such a complex job? What are these nanocarriers actually made of, and how do they ensure they're hitting the right targets within the body's intricate defense system?
It truly is a remarkable feat, a brilliant fusion of advanced materials science, cutting-edge engineering, and deep immunological understanding. The design process is meticulous, involving not just choosing the right nanocarrier, but also selecting the perfect antigens, the cancer's unique identifiers, and, critically, ensuring they are presented to the immune system in the most effective and natural way possible. It's like designing a custom-built, highly intelligent drone that knows exactly what to carry, where to go, and how to deliver its message.
Okay, so let's start with the vehicles. What are the options?
Right, let's start with A nanocarriers, NCs, the smart delivery vehicles. These are the workhorses, the sophisticated couriers of the immune system, and their material matters immensely. First, we have polymeric nanoparticles. A very common and popular choice here is polylactic co-glycolic acid, or PLGA. It's highly favored because it's biocompatible and biodegradable, meaning it breaks down harmlessly into metabolizable lactic and glycolic acid modules within the body, which are then naturally cleared.
So it's safe.
Yes, generally considered safe. What's more, its size, stability, and even its solubility can be precisely fine-tuned during manufacturing. PLGA is incredibly versatile, capable of encapsulating a wide range of cargo, including both hydrophobic and hydrophilic drugs, peptides, and even nucleic acids like mRNA or siRNA. While incredibly promising and able to induce protective immunity, they do come with some challenges. For instance, they can sometimes aggregate, clump together, which can affect their performance. Their cellular adhesion can sometimes be low, and their manufacturing process can be quite complex.
Ah, the practicalities.
Exactly, often involving organic solvents, which can limit their large-scale production and clinical translation. But research continues to overcome these. Then there are inorganic nanoparticles, which offer some truly exciting multifunctionality that goes beyond mere delivery.
Like what?
Well, quantum dots, QDs for example, possess unique fluorescence properties. This allows researchers to dynamically track and image the nanovaccine's journey in real time within the body, for instance, seeing where they go in lymph nodes. Gives invaluable insights.
So you can actually watch them work.
Pretty much, yeah. Then gold nanoparticles have shown remarkable potential. Imagine gold glycan nanoparticles precisely engineered to incorporate a listeriolysin O peptide. That's a bacterial component that helps antigens escape from cellular compartments, alongside powerful immune checkpoint inhibitors like anti-PD-1 or anti-CTLA-4. This isn't just theoretical. In striking melanoma mouse models, this ingenious combination has led to a staggering 97% to 98% regression of tumor volume.
Wow, 98% regression. That's incredible.
It is truly impressive and points to their immense therapeutic potential. And magnetic nanoparticles can be used in incredibly clever ways to literally manipulate dendritic cell migration using external magnetic fields. You can guide these crucial immune cells to where they're needed to enhance anti-tumor efficacy, almost like a microscopic GPS, directing the intelligence officers to the training ground.
That sounds like science fiction.
It does, doesn't it? However, despite their clear advantages like low toxicity, hydrophilicity, and high stability compared to organic materials, a significant hurdle for inorganic nanocarriers is their detection by immune cells. They're often rapidly phagocytosed or engulfed by these cells and shunted away for disposal. This complicates their journey towards FDA approval for cancer treatment, even with all the strong preclinical data supporting their efficacy.
So their visibility is a double-edged sword.
Kind of, yes. Another fascinating category is biomimetic nanocarriers. These are incredibly clever because they are vesicles derived directly from natural cell membranes from sources like tumor cells, platelets, or even red and white blood cells. The genius here is that they retain the intricate characteristics of the original cell, including all the proteins and glycoproteins present on their surface.
So they look like cell.
Exactly. This offers significant advantages, such as homotypic targeting, meaning they naturally gravitate towards where the original cell would. Immune evasion properties, because they look normal to the body, and longer circulation times in the bloodstream, allowing them more time to reach their target. They often demonstrate improved performance over bare nanoparticles because they harness these natural biological properties.
Very clever. And you mentioned VLPs.
Yes, virus-like particles,
Yeah.
VLPs. These are essentially non-replicating viral shells, empty protein cages that perfectly mimic a virus's outer structure, but without any of the infectious genetic material. Because they look so much like a real virus, they are highly effective at triggering robust immune responses, as our immune system is naturally primed to respond to viral structures. You might be familiar with them already, as they have been successfully used in approved preventive vaccines, such as Gardasil and Cervarix for HPV.
Right, the HPV vaccines. Okay, so there's an entire arsenal of choices when it comes to the delivery vehicle. But regardless of the material, I imagine size is a huge factor, right? You can't just make them any size you want.
You're absolutely right. Size matters for delivery immensely when designing these nanocarriers. For effective delivery to the lymphatic system, which is essential for priming a strong immune response by interacting with dendritic cells in the lymph nodes, particles between 10 and 100 nanometers show the highest lymphatic uptake efficiency.
10 to 100 nanometers, that's tiny.
Incredibly tiny. Think of it as a specific size window for optimal entry into the immune system's command centers. If they're smaller than 5 nanometers, they tend to be quickly cleared by the bloodstream, flushed away before they can do their job. And if they're larger than 200 nanometers, their accumulation in lymph nodes significantly decreases, making them less effective at reaching the T-cell training grounds. So it's a very precise sweet spot for these nanocarriers.
Okay, so we have the vehicle, perfectly sized. Now, what about the cargo? Finding the cancerous fingerprint.
Exactly. Now let's move to B, antigen selection and presentation, finding the target. This is where we identify cancer's unique fingerprint, the mugshot our immune system needs. The most exciting targets are neoantigens. These are not just any tumor antigens. They are unique, tumor-specific antigens that arise from genetic mutations within the cancer cells. Things like single nucleotide variations, SNVs.
A single letter change in the DNA code.
Precisely. Or insertions deletions, INDLs, when genetic material is added or removed. These are highly personal to the patient's tumor. What's particularly compelling is that INDLs, for instance, can create entirely new open reading frames, known as neo-ORFs, which can lead to fundamentally new proteins. These often generate an even higher proportion of neoantigens per mutation compared to simple SNVs, making them particularly potent targets.
More bang for your buck, mutation-wise.
You could say that, yeah. To find these unique signatures, we use what's called a reverse immunology approach. It's a bit like a detective story, but with a lot of high-tech genomic tools. Researchers sequence a patient's tumor tissue and then compare it to their normal tissue to precisely identify these genetic mutations that are unique to the cancer. Then, sophisticated computational tools come into play, predicting which of these mutations might lead to neoepitopes, the specific peptide fragments, that can be effectively presented on MHC Class I molecules to alert T cells.
So we can actually find these unique cancer signatures in a patient then?
Yeah.
Are there real-world examples of this working? Because it sounds incredibly complex.
It is complex, but we've seen some truly remarkable real-world success stories. Consider the KRAS G12D mutation study as a groundbreaking example. In a study published in Science, researchers successfully applied this strategy to a patient with metastatic colorectal cancer. They had identified T cells from this patient that were specifically reactive against a mutant KRAS G12D antigen, a common mutation in many cancers. So they went further. They resected lung lesions from the patient, meticulously sequenced the tumor, and found 61 distinct mutations. Then, they cleverly used tandem minigene, TMG constructs, essentially engineered snippets of RNA encoding these mutated proteins, which were electroporated directly into the patient's own dendritic cells.
Into their own DCs.
Yes, their own DCs. This ingenious step was crucial because it ensured that the peptides were processed and presented naturally by the DCs, just as if they were made by a tumor cell. The subsequent adoptive transfer of these specifically reactive T cell clones resulted in objective regression of all lung metastases.
All of them. All of them.
All of them. This is a powerful proof of concept demonstrating that you can identify and target these unique cancer signatures to achieve a significant clinical response.
And that highlights the importance of getting the presentation right, doesn't it?
Absolutely. This Tran et al. Study highlighted something absolutely crucial: the importance of ensuring natural processing. Many experimental systems can elicit T-cell responses to epitopes that aren't processed naturally by the body, meaning they wouldn't work therapeutically in a real patient. To get it right, methods like electroporating mRNA that encodes the full-length protein into antigen-presenting cells, APCs, mimicking how endogenous proteins are made.
Right, like the TMGs they use.
Exactly. Or pulsing APCs with long peptides that require cellular processing, mimicking how exogenous proteins are handled, are considered the most effective ways to ensure this natural pathway is mimicked. Just pulsing with short 816er peptides, which are minimal epitopes, can be misleading because it doesn't guarantee natural processing by the immunoproteasome, or correct transport by TTI, or precise trimming by ERAP1. The immune system is very discerning, and if the antigen isn't presented exactly right, it won't recognize it as a threat.
Okay, so natural presentation is key. Finding these neoantigens seems like a huge task, though.
It is, but that's where C, the power of prediction in silico insights, comes in. This is where computation meets biology, allowing us to accelerate this discovery process. We have incredibly sophisticated computational tools and databases today, like NET-MHC-PAN and the IEDB, Immune Epitope Database. These algorithms can predict which peptides are most likely to bind to specific HLA molecules with high affinity, literally sifting through millions of possibilities to find the needles in the haystack.
How accurate are these predictions?
They're getting better all the time. One common key metric in this predictive process is the IC50 value, which measures how strongly a peptide binds to an MHC molecule. A predicted MHC binding IC50 of 500 nanomolars or less has been shown to have about 90% sensitivity for identifying true T-cell reactive epitopes. Another useful metric, especially when predicting across different HLA types, is the percentile rank, which helps avoid bias towards certain HLA alleles and allows for comparison across different individuals.
Interesting. But does strong binding always mean a good target?
That leads us to the paradox of weak binders. This is a truly intriguing observation in immunology. A significant proportion of validated T-cell antigens, the very targets our immune system can recognize and fight, actually show weak predicted binding in these algorithms.
Weak binding. How does that work?
Well, their IC50 is greater than 500 nm, sometimes even exceeding 10,000 nm. One compelling theory for this is that these weakly immunogenic antigens haven't been subjected to intense immunoediting by the immune system. In other words, the immune system hasn't yet selected against them because they were less visible initially, less likely to trigger a strong immediate response. This then allows them to become the dominant targets after other, more obvious or strongly binding targets have been eliminated by conventional therapies or the initial immune response.
So the less obvious targets might actually be the better ones later on.
Potentially, yes. It's a testament to the dynamic, evolving interplay between the tumor and the immune system, revealing a crucial blind spot in cancer's defense. The very antigens it didn't try to hide might be its ultimate undoing, making them prime targets for our new vaccines.
Fascinating. Have these prediction methods led to successful vaccines in practice?
Yes, we've seen incredible success in melanoma vaccines using these predictive approaches. In the development of personalized melanoma vaccines, researchers meticulously use prediction algorithms like MHC to identify up to 30 high-affinity, patient-specific MHC Class I restricted binders derived from mutant peptides. They prioritized neo-ORFs and mutations in key binding regions, and then incorporated up to 20 putative neoantigens into personalized vaccines for each patient. The results were truly remarkable. Out of 6 vaccinated patients, 4 were recurrence-free 25 months after vaccination. A remarkable achievement for a cancer known for its aggressive recurrence.
25 months recurrence-free is huge for melanoma.
It really is. And the remaining 2 experienced complete tumor regression after additional immune checkpoint therapy. This really shows the incredible potential of combining precision antigen identification with modern immunotherapy, moving us ever closer to truly personalized cancer treatment.
So we've got these incredibly precise nanocarriers, and now we know how to find cancer's unique fingerprints, those neoantigens, using powerful computational tools. But it's one thing to have the map, it's another to have a guided missile. Here's where it gets really interesting. How do we make sure these nanovaccines hit their mark directly, especially those all-important dendritic cells we talked about earlier? How do we ensure they deliver their critical message to the right place for maximum impact?
This is truly a crucial area of innovation, and one where we're seeing some fantastic breakthroughs, because getting the message to the right recipient is everything. Remember, dendritic cells are the immune system's primary intelligence officers, the ones responsible for effectively capturing, processing, and presenting antigens to prime T cells. Getting the nanovaccine directly to them significantly amplifies and enhances the entire immune response. It's like bypassing all the intermediaries and going straight to the top, ensuring the T-cell army gets its marching orders loud and clear.
Okay, so how do you target DCs specifically?
One incredibly promising strategy involves using specific DC-binding peptides, DCBPs. These are small peptides, tiny protein fragments that have a high affinity for specific receptors found directly on the surface of dendritic cells. They're actually more efficient than using larger molecules like whole antibodies because their small size helps them cross biological barriers much more easily, making their delivery very precise and efficient. They are literally the homing beacon.
Small and targeted. Makes sense.
Let me tell you about a fascinating case study from Zhejiang University in China. Researchers there conducted a brilliant study where they engineered a nanovaccine using virus-like particles, or VLPs, derived from porcine circovirus type 2, or PCV2. This wasn't just any VLP. They cleverly decorated these VLPs with different DC-binding peptides, effectively turning them into targeted delivery systems. And one particular peptide, which they called DCBP3, showed exceptional results, truly a standout performer in its ability to target dendritic cells.
So they tested different homing beacons.
Essentially, yes. And the cleverness of their design extended to using a flexible linker, specifically glyglycer repeats, between the PCV2 capsid protein and the DCBPs. This seemingly small detail is incredibly important because it ensures that the peptides can bind properly to the dendritic cell receptors without any steric hindrance, allowing the VLP to assemble correctly and present the antigens optimally. It's a bit like making sure the GPS receiver has a flexible antenna to get the best signal.
Right, the little details matter. Did it work well?
What they found was truly impressive. The engineered VLPs were remarkably stable, maintaining their morphology and homogeneity even after 3 to 6 months of storage at 4 degrees Celsius, which is vital for practical vaccine development. Over 50% of the VLPs remain soluble, showing excellent shelf life.
Good stability, that's important.
Very. When they tested these in lab-grown, murine, bone marrow-derived dendritic cells, the capped DCBP3 VLPs showed a significantly stronger uptake by these cells, with much brighter fluorescent signals compared to other DCBPs they tested. This unequivocally means the DCBP3 was really doing its job of targeting and delivering the VLPs directly to the dendritic cells.
So the targeting worked? Did it activate the DCs?
Yes. This enhanced uptake then translated directly into increased dendritic cell activation and boosting DC maturation. The capped DCBP3 VLPs significantly upped the expression of key maturation markers like MHC2, CD80, and CD86, along with the cytokine IL-6. This is exactly what you want to see. It showed that the dendritic cells were indeed getting ready for action, preparing to process antigens and prime the immune response effectively. They were essentially being activated to their full potential.
And did that translate into a better immune response overall?
It certainly did. It led to potent humoral immunity. In mice, the capped DCBP3 VLPs elicited significantly higher levels of CAP-specific IgE antibodies. In fact, they were 1.55 times higher than even a commercial vaccine they used for comparison. This indicates a much stronger antibody response. It also boosted specific antibody subclasses, IgG1 and IgG2A, by 1.2-fold and 1.8-fold respectively over the commercial vaccine at different time points.
Impressive numbers.
Even more importantly, they increased PCV2-specific neutralizing antibodies by 2.5 times compared to untargeted VLPs, reaching or even surpassing the levels induced by commercial vaccines. Neutralizing antibodies are absolutely crucial because they directly block pathogens from infecting cells, providing a very strong and protective defense.
So, better antibodies. What about the T cells?
Beyond antibodies, the capped DCBP3 targeted vaccine also triggered a powerful, robust cellular immunity. It induced high levels of lymphocyte proliferation in splenocytes, those are immune cells from the spleen, indicating that T cells were rapidly multiplying to fight the perceived threat. Furthermore, it significantly increased the production of key cytokines like IL-2, IL-10, and IFN. IFN is critical for activating cells that kill intracellular pathogens, including cancerous cells, and orchestrating a strong anti-tumor response.
Right, interferon gamma, crucial for cell killing.
Exactly. Meanwhile, IL-2 is a potent immunostimulatory molecule that helps T and NK cells activate and expand, and it can establish robust memory cell populations, which is vital for long-term protection against recurrence. This strong cellular response is essential for truly clearing out infected or cancerous cells.
So it's not just about getting the antigen to the immune system, but directing it to the right cells, those critical dendritic cells, with incredible precision. And this fascinating research from Zhejiang University absolutely shows that DC targeting is a game changer. It's like sending a highly trained scout to relay intelligence directly to the commander.
Exactly. This research strongly supports the use of DC-binding peptides as a powerful tool to further boost antigen presentation in dendritic cells. This has major implications for enhancing the efficacy of VLP-based and nanoparticle-based vaccines, not just for viruses, but for cancer too. It's a fantastic example of innovative global research pushing the boundaries of what's possible in medicine, and it truly provides a blueprint for how we might create more effective and targeted cancer nanovaccines. The precision targeting here is a monumental step forward.
This all sounds incredibly promising, like we're on the verge of a true revolution in cancer treatment. But where do we stand with cancer nanovaccines in the clinic right now? And what are the big challenges still ahead that researchers and clinicians need to overcome before these breakthroughs can reach every patient who needs them?
You're right, the promise is immense, and we've seen significant progress, but there are certainly formidable hurdles to overcome before these nanovaccines become widely available and universally effective. First, one of the biggest obstacles for any cancer immunotherapy is A, conquering immunosuppression in the tumor microenvironment, TME. The tumor is incredibly cunning. It doesn't just grow, it actively creates an immunosuppressive environment around itself, a kind of defensive shield or cloaking device that effectively shuts down the very immune cells trying to fight it. Think of it as the tumor's way of hitting the mute button on the immune system.
Right, that TME is a major barrier. How can nanovaccines help there?
Well, nanovaccines are being brilliantly designed to address this in several clever ways. They can be combined with immune checkpoint inhibitors, which are revolutionary drugs like anti-PD-1, PD-L1, and anti-CTLA-4 antibodies. These drugs essentially take the brakes off T cells, allowing them to activate and attack cancer. What's even more advanced is that while these are usually administered separately, researchers are now exploring engineered nanovaccines that directly encode these inhibitors or carry them as cargo.
So the vaccine itself delivers the break-releasing drug.
Exactly. This means they can be delivered right to the tumor site, maximizing their local impact while potentially minimizing systemic side effects, which is a huge step forward in reducing toxicity. Furthermore, nanovaccines can also deliver other potent agents, such as small molecule inhibitors or siRNA, small interfering RNA molecules.
siRNA, that's the gene silencing tech.
Yes, it's like a tiny genetic switch that can silence specific genes. These are specifically designed to shut down genes that contribute to the TME's suppressive nature, effectively dismantling the tumor's mute button. This targeted delivery not only protects the cargo from degradation, but also extends its half-life, meaning it stays active longer, further enhancing the anti-tumor immune response directly where it's needed most. It's a multi-pronged attack on the tumor's defenses.
So it's not just about activating the immune system, but actively disarming the tumor's countermeasures. That's clever.
What's the real-world status of these nanovaccines today? Are they just in labs, or are they moving towards patients?
They are definitely moving towards patients, and it's a rapidly evolving field. Looking at the current clinical landscape, we can see a lot of activity. It's worth noting that we already have some approved cancer vaccines, though not all are nanovaccines in the strictest sense. For instance, TICE, BCG, a bacterium, has been used for decades to treat early-stage bladder cancer.
Right. BCG for bladder cancer. And Sipuleucel T, known as Provenge, is an autologous cellular immunotherapy for metastatic prostate cancer, where a patient's own immune cells are trained outside the body and then reinfused. And of course, the incredibly successful preventative vaccines like Cervarix, Gardasil, and Gardasil 9 protect against HPV-related cancers.
The prevention ones are key too.
Absolutely. The real excitement, however, is in the burgeoning number of nanovaccines in clinical trials. In phase I trials, which focus on safety and dosage, we're seeing trials underway for lipid nanoparticle, LNP, encapsulated mRNA vaccines for Epstein-Barr virus, EBV, positive advanced malignancies, like a significant trial at West China Hospital.
LNP, that's the fat bubble delivery system?
Exactly, the lipid nanoparticle. It's a tiny protective fat bubble that encapsulates delicate mRNA or other nucleic acids, shielding them until they reach their target cells. There are also personalized neoantigen peptide vaccines and RNA lipid particle, RNA-LP, vaccines for glioblastoma, a very aggressive brain cancer. In these trials, tumor material is even collected post-surgery to create patient-specific vaccines, demonstrating true personalized medicine in action.
Wow, tailoring it right from the patient's own tumor.
Precisely. Moving to phase 2 trials, the focus shifts to efficacy and immune response in larger patient groups, refining dosage and schedules. Studies are evaluating powerful combinations, such as the DHER2 plus AS15SI, which is a recombinant protein targeting the HER2 protein, combined with an adjuvant system AS in a liposomal formulation.
So targeting HER2 wrapped in a lipid bubble with immune boosters.
You got it. Combined with lapatinib for HER2 overexpressing metastatic breast cancer. Other trials involve various liposomal vaccines for non-small cell lung cancer and HPV-related cervical cancer, often strategically combined with conventional treatments like chemotherapy or radiation to find synergistic effects that could enhance patient outcomes.
Combining old and new strategies.
Yes, finding the best combinations. And finally, in phase 3 trials, this is the last and largest stage before potential approval, where efficacy is confirmed against standard therapies. Promising trials include V940, an individualized neoantigen therapy delivered in an LNP formulation combined with pembrolizumab, a known immune checkpoint inhibitor for high-risk melanoma and non-small cell lung cancer. Another significant trial involves a combination of an adenoviral vector and an LNP-formulated self-amplifying mRNA for metastatic colorectal cancer. These trials represent large-scale international efforts aiming to definitively prove the efficacy of these new approaches and bring them to market.
That's a staggering amount of activity, suggesting this isn't just a pipe dream. But beyond clinical trials, what are the inherent difficulties in translating this cutting-edge science from the lab to widespread patient access? What are the biggest hurdles?
Indeed, the challenges are significant, but certainly not insurmountable, given the ingenuity involved. One major biological complexity is how nanoparticles interact with the body's biological components once they're inside. When nanoparticles enter the bloodstream, they quickly get coated by a biomolecular corona made up of proteins, lipids, and carbohydrates from the blood.
A corona? Like a halo around the particle?
Kind of, yeah. This corona can profoundly impact how the particles are distributed, how they're taken up by cells, and ultimately their overall therapeutic efficacy. It can essentially change their identity and behavior. To overcome this, we desperately need standardized characterization methods and more representative experimental models in preclinical settings. Models like organs on a chip, human on a chip, and three-dimensional spheroids are proving vital.
Better lab models.
Exactly. Mimicking living systems more accurately than traditional cell cultures, allowing for better prediction of human response. Artificial intelligence is also being developed to integrate data from these complex models for more robust simulations, accelerating development and reducing reliance on animal testing.
Okay, biological complexity. What else?
Then, there are significant challenges with manufacturing and scalability. Moving from small batch lab production to large scale, consistent manufacturing for millions of patients is incredibly tough. Ensuring batch-to-batch reproducibility and consistent quality requires adhering to strict good manufacturing practices, GMPs, and overcoming the inherent variability in complex nanostructures.
Making them the same every single time on a massive scale.
Precisely. Researchers are actively exploring autonomous manufacturing processes and using AI to optimize synthesis parameters and monitor quality in real time. This could drastically improve efficiency, consistency, and reduce human error, making mass production viable.
And the cost must be huge, too.
Absolutely. Costs and accessibility are major considerations. Nanomedicines, especially personalized ones, can be incredibly expensive due to the intricate design and manufacturing processes. Simplifying processes, scaling up production, and automating elements are key to making them more affordable and widely accessible, which could significantly impact global healthcare by providing more effective treatments to more people, especially in underserved regions. This is a global health challenge that requires innovation in both science and economics.
Right, it needs to be reachable for everyone.
And finally, patient variability presents a critical challenge that speaks to the very core of personalized medicine. Immune responses vary significantly among patients due to individual genetics, lifestyle, and prior medical history. Individuals who have undergone prior treatments like extensive chemotherapy might have impaired immune systems, which could significantly affect vaccine efficacy.
So the patient's history matters a lot.
It really does. For personalized nanovaccines, establishing standardized customization protocols and reducing the production time from tumor biopsy to vaccine delivery are crucial for timely clinical implementation. Furthermore, more precise machine learning models are needed to reliably predict immunogenic neoantigens for each individual patient, making the process even more efficient and accurate, ensuring that each patient receives the most effective, tailor-made treatment.
So what we're witnessing is a transformation in how we approach cancer, moving from broad, often harsh attacks to incredibly precise, highly personalized immunotherapy. It's a fundamental shift in strategy, almost like moving from carpet bombing to surgical strikes at a cellular level.
Indeed. Cancer nanovaccines, through their precise engineering and remarkable ability to teach our own bodies to recognize and eliminate these elusive cancer stem cells, truly offer an incredibly hopeful and powerful path forward. The ongoing research and the myriad of clinical trials are pushing the boundaries of what's possible, not just in treating existing tumors, but in preventing the very recurrence and metastases that have long plagued oncology. We are genuinely on the cusp of empowering the body's own defenses to win the long war against cancer. And, just imagine what new frontiers interdisciplinary collaborations will unlock when we fully integrate things like artificial intelligence, multiomics, and synthetic biology to design the next generation of nanovaccines. We could move beyond even what we've imagined in precision medicine and patient stratification, truly customizing health solutions right down to the individual's unique biological fingerprint. It's a really exciting time.